So thank you for the introduction, Alice. Hi, my name is Kira, and I am a fourth-year PhD student at the University of Illinois Urbana-Champaign, and today I will be presenting part of my dissertation research on the hybridization dynamics and population genomics of a Manacus hybrid zone. A well-known example of a hybrid is the useful mule, which is the offspring of a horse and a donkey. But what exactly is a hybrid? A hybrid is the offspring of two different species, most often talked about in the context of the biological species concept, where two separate species form hybrid offspring that are either inviable or infertile, like the mule, indicating the parents are indeed separate species. However, beyond the biological species concept, hybridization is an important evolutionary process. Hybridization is a natural laboratory to study speciation, what forms species boundaries, what breaks species barriers, and how novel genotypes admix in hybrid zones. A hybrid zone is a geographic area where two species meet and intermix. Hybrid zones are dynamic and they can change over time. The parental species could continue to diverge and they could develop more reproductive barriers. They could have reproductive barriers break down and collapse back into a single species, or they can remain stable over an indefinite period of time. My research focuses on an avian hybrid zone of two manakin species in Western Panama. Manakins are a neotropical lekking bird, as many of you know, that have communal breeding grounds where the males make and defend territories and females choose a male to mate with, and then the females do all the parental care themselves. Due to this type of mating system, there's strong sexual selection driven by female choice in the system, making it very interesting to look at for hybrid zones, which I'll get into in a moment. So here we have our white-collared manakin, one of the parental species. Notice the white collar and yellow belly. The golden-collared manakin with this golden collar and green belly. And here we have our hybrid, who has this yellow throat and yellow belly, kind of mixing both the parents. Then here you have our important females, who are just drab green in all the different morphs. So the females are very important because in previous studies, it's found that the females may prefer males with more yellow plumage than this white plumage. In addition, the hybrids were found to be more aggressive, so that might get them better placement on the lek, further leading to improved mating success. So there might be some selective advantage in this hybrid zone for having yellow versus a white throat. However, there's a lot of variation, phenotypic variation in this hybrid zone. So all three of these males are hybrid males, but they're very clearly, they all look different. They have different combinations of what the parental species look like. So this is showing that these individuals have very different genetic admixtures from their parental species. This hybrid zone is relatively well studied and it was originally characterized about 25 years ago. Here's a map of the hybrid zone in Western Panama. We have the white collards up here going into Costa Rica, our golden collards down here back towards the rest of Panama, and this area in the middle for our hybrids. These black dots are my sampling sites, which are mimicking and following the original transect from the 90s. From previous research, the genetic center of the hybrid zone was determined to be around sampling site 9 here, meaning the birds here were overall the closest to a 50-50 genetic admixture of each parent. But the phenotypic transition, where you have birds with yellow plumage on their throats, which changes to white plumage on their throats, happens here. So based on these two different transition points, it's hypothesized that there's asymmetrical introgression of yellow plumage coming from the golden collared manakins and introgressing into white collared manakins because it might have a selective advantage. But if there's strong sexual selection for this male trait, then this prompts the question, is this trait continuing to spread? Based on female preference and sexual selection alone, we would expect this beneficial trait to continue spreading into the white collared population. However, without a second time point, we don't know what this yellow plumage trait is doing, and we don't know what the genetics are doing. What are the spatial and temporal dynamics of this hybrid zone with strong sexual selection for an introgressing trait? And this is where my dissertation is coming in. So I'm going over one of my objectives today, where I'll be investigating the spatial and temporal dynamics of this hybrid zone. First, in order to see what this is doing, I have to look at the current genetic population structure of this hybrid zone in order to compare it to the original study to see how it has changed over time. Overall, this lets me determine just, is this hybrid zone moving? And if it is, how far has it moved? So I went out to Panama over the last 4 years, caught 600 manakins from all across the hybrid zone, getting 600 genetic samples to thoroughly represent each population in this transect. I captured manakins using mist nets And I also, as part of my dissertation, did net searching, radio telemetry, testing for avian malaria, and feel free to message me about those results later, but I don't have time to go over that. Hopefully next conference I'll talk about that. So to genotype, I used RAD sequencing, or restriction site associated DNA sequencing, which, what does that mean? So we're using restriction enzyme sites to kind of sample the genome, and we're only sequencing like the sections around these cut sites. So we get a sampling of the genome, but we get a large quantity of consistent markers in order to look at a lot of burns with like much lower cost of sequencing. So here we can see, as Jacob actually introduced, this is a fine RAD structure plot, which he introduced excellently. So here's the general genetic structure of 68 of the individuals. We have the my individuals down here, and they form this matrix of co-ancestry with the blue and darker colors meaning more like related, and then the yellow being less related. The biggest takeaway from this plot is that I have highlighted here, there's two main blocks that are grouping together. We have the golden collared hybrid population with golden collared population and 1 hybrids here, and then all the other hybrids are grouping generally with the white collared populations. But let's look at this further. So this is a hybrid index of these same individuals with the golden collared mannequins on the bottom left and the white collared mannequins on the upper right, with each of the hybrid populations in the center. This hybrid index is showing generally how many of the alleles or what portion of the genetics of these hybrids comes from one parent or the other. So the biggest thing that jumps out from this graph is shown in the population here. We can see that there's a large amount of variation in the hybrid index in this population in particular, highlighting this one as a possible genetic center of the hybrid zone. But let's take a closer look at this by looking at the geographic lines. So a quick note on geographic lines. When we're looking at geographic lines between two species, you expect to find one of these, generally one of these options. On your x-axis, you have the literal physical distance between populations, and then on the y-axis, you have your allele frequency. So whether it is more like one mannequin species or the other. On the left here, you have a neutral cline that's mostly governed by isolation by distance, or how far apart these mannequins are, where these alleles are kind of just drifting between the two populations, but nothing much going on. On the right here, you have some kind of selection, you can't tell which kind, some kind of selection where you see the switch transition from one species to another in the allele frequency. So these are kind of what we're looking at when we look at our clines. So here is a graph of some randomly selected fixed difference alleles between the two parental species from my first 68 samples. In black, I made an approximation of a geographic cline of one of the previous studies. And in red, we have plotting of 50 randomly selected alleles from my study. And overall, you can see that the majority of these alleles are following the previous transect. They are swapping over pretty consistently altogether here, indicating to me that this hybrid zone is probably not moving. But let's take another look at the specifically alleles from the Z chromosome. And so here what's even more striking is it's actually even more narrow. This is also 50 alleles that are so tightly moving together that it doesn't even look like 50 alleles switching over, that are again crossing over about this half and half admixture mark at around the same place. So overall, what have we found? Is this hybrid zone moving? No, it is actually not. We found that the genetic center of the hybrid zone is in the same place at this population 9 as it was in the 90s. And then we also found, while I was sampling through the transect, that the phenotypic transition is also about here at this river. Although I did find 1 or 2 white collared or yellow collared mannequins over on this side, the phenotypic transition and the genetic transition are about in the same place. So it appears that even with strong sexual selection, possibly pushing the introgression of this yellow plumage trait, the hybrid zone has somehow remained stable over time. So the next question I want to address in this dissertation is, why is the hybrid zone stable? What are the mechanisms controlling fertility in this hybrid zone? So I'm looking at things such as reproductive success, survivorship, egg viability, and the incidence rates of avian malaria. So stay tuned for those results coming up at the next conference. And I would like to acknowledge my wonderful advisors, Jeff Braun and Mike Braun, Braun Squared, and my lovely field assistants, the Studying Tropical Research Institute, funding sources. And I would love to take any questions.